In December 2022, a team led by Eske, Willerslev, and Kjaer announced the recovery of authentic 2-million-year-old environmental DNA from permafrost sediments at Kap Kobenhavn in northern Greenland, shattering previous age records and redefining what is technically possible. Molecular traces revealed a vanished forest ecosystem, ancestral megafauna, and Arctic temperatures up to 19 degrees warmer than today, all preserved in ordinary clay and Smectite minerals. Yet the DNA never fossilized. It adsorbed onto mineral surfaces, surviving deep freeze and time. Official climate models cannot fully explain these findings, raising urgent questions about how polar regions respond to carbon dioxide and what ancient genes could offer for future resilience. The sequence of discovery, preservation mechanism, and groundbreaking implications begins here. In December 2022, a paper in Nature by Eske Willerslev's group established a new benchmark for ancient DNA recovery, 2 million years. This record nearly doubles the previous maximum, which stood at 1.2 million years from Siberian mammoth remains. Sediments from Kap Kobenhavn, drilled in northern Greenland, yielded environmental DNA that predates the earliest known permafrost genomes by over 800,000 years. The team's findings were independently verified, and the raw sequence data were deposited in the European Nucleotide Archive under accession PRJ EBI 55522. Willerslev, a professor at the University of Copenhagen and the University of Cambridge, led the study with Kjaer. Their work was supported by the Lundbeck Foundation and involved more than 40 researchers from Denmark, the UK, and Sweden, reflecting an international collaboration. The announcement drew immediate attention from the scientific community because no authentic DNA had previously been recovered from sediments older than the early Pleistocene. The study's age assignment relied on magnetostratigraphy, biostratigraphy, and uranium-thorium dating, placing the Kap Kobenhavn Formation in the late Pliocene from 2.1 to 1.95 million years ago. The Nature paper outlined the authentication protocols, including cytosine deamination analysis and replication in independent laboratories. The headline result, recovery of DNA 2 million years old from ordinary permafrost, challenged assumptions about molecular decay limits. Willerslev described the outcome as a technical and conceptual leap, saying the project pushes the boundaries of what ancient DNA can reveal. The age record set at Kap Kobenhavn now frames the central scientific puzzle, how DNA molecules could persist for 2 million years in Arctic sediment, and what this means for reconstructing lost ecosystems and climate histories. Kap Kobenhavn sits at 78 degrees north latitude on Greenland's northern coast within Pearyland. The site's coordinates place it only 900 kilometers from the North Pole, in a region now dominated by barren permafrost and exposed bedrock. The formation itself is a sedimentary archive, built up over 20,000 years during the late Pliocene, as confirmed by magnetostratigraphy and biostratigraphic markers. Its stratigraphy consists of silty clays and fine sands, deposited in a shallow marine embayment at the edge of a coastal plain. The sedimentary sequence reaches nearly 100 meters in thickness, preserving a continuous record of environmental change across multiple layers. Kjaer, a stratigrapher at the University of Copenhagen, led the geological mapping and core analysis. His team identified distinct horizons within the formation, each marked by shifts in grain size, mineral content, and organic matter. These layers provide a physical timeline, anchoring the site's age between 2.1 million and 1.95 million years. Magnetostratigraphic work established the sequence's polarity pattern, linking it to global geomagnetic reversals and allowing precise correlation with the geologic timescale 2020. The Kap Kobenhavn archive is undisturbed by glacial erosion, making it one of the most complete Pliocene sediment records in the Arctic. Its stratigraphic continuity is essential for interpreting how DNA molecules persisted in situ, shielded by permafrost and mineral matrices. The site's sedimentary architecture underpins every subsequent analysis, from molecular extraction to ecosystem reconstruction. DNA molecules, once released from ancient cells, are exposed to rapid chemical breakdown. Yet, in the Kap Kobenhavn sediments, fragments as short as 30 to 50 base pairs have persisted for 2 million years. The critical factor is adsorption to mineral surfaces, particularly Smectite, clay, and quartz. Laboratory experiments using salmon sperm DNA demonstrated that Smectite binds DNA with greater capacity than non-clay minerals. Extraction yields from clays averaged around 7%, confirming a strong affinity between DNA and the layered structure of Smectite. Quartz, which accounts for nearly half the sediment by weight, also retains DNA, but with slightly lower efficiency. X-ray diffraction analyses of Kap Kobenhavn Cores identified Smectite concentrations between 1.2 and 3.7% by weight, with total clays reaching up to 9.6%. Scanning electron microscopy revealed DNA adhering to the surfaces of microscopic mineral grains, forming a protective barrier against enzymatic attack. The marine depositional environment, with its neutral pH and low ionic strength, further promotes electrostatic binding between the DNA phosphate backbone and mineral active sites. At a mean annual temperature of minus 17 degrees Celsius, thermal age modeling shows that DNA decay rates are slowed by a factor of over 700 compared to conditions at 10 degrees Celsius. This molecular vault effect, unique to Smectite-rich permafrost, explains the survival of DNA far beyond previously accepted limits. The plausibility of 2 million-year-old genetic material is grounded in the combined protection offered by cold, stable burial, and mineral adsorption, setting the stage for the extraction and sequencing protocols that follow. Each Kap Kobenhavn sediment core was drilled, labeled with a unique alphanumeric code, and immediately logged for temperature using digital recorders. Field teams operated under strict chain of custody procedures, documenting every transfer from permafrost extraction to laboratory freezer. Core sections were sealed in vacuum-insulated polymer bags and stored at sub-zero temperatures throughout shipment to Copenhagen. Laboratory staff wore sterile gowns, masks, and gloves. All surfaces and tools were decontaminated with bleach and UV light before core opening. Each sediment sample was processed in a dedicated clean room with negative pressure airflow and HEPA filtration to minimize airborne contamination. Blank extraction controls accompanied every batch, using sterile water and empty tubes to detect laboratory or reagent-derived DNA. Library preparation included unique molecular barcodes for each sample in control. All steps, from sediment subsampling to final sequencing, were logged in a digital ledger, with cryptographic hashes ensuring traceability. Sequencing reads from blank controls were analyzed separately. Their taxonomic profiles showed only common laboratory microbes at low abundance. Statistical modeling with a Poisson estimator calculated the probability that mastodon and other megafaunal reeds originated from ancient sediment rather than contamination. The result exceeded 99.9% confidence for ancient origin. Data integrity was further supported by independent replication in Oslo and by the deposition of raw sequences in the European Nucleotide Archive under accession PRJEB 55522. DNA, extracted from Kap Kobenhavn, sediments identified over 70 genera of plants, representing a diverse forest ecosystem at 78 degrees north latitude. Pollen spectra and macrofossil fragments confirm the presence of poplar, birch, willow, and thuja, trees that today do not grow anywhere in Greenland. Shrubs such as alder, spruce, and northern white cedar were also detected, alongside aquatic plants, sedges, and grasses. This composition matches a boreal temperate forest, not the current polar desert. Quantitative analysis showed that 39% of the detected plant genera are absent from modern Greenland flora. The high diversity and presence of temperate taxa indicate a much warmer climate during the late Pliocene, with mean annual temperatures reconstructed as 11 to 19 degrees Celsius above present values. The floral DNA was authenticated by cytosine deamination patterns and matched against modern and ancient reference libraries. The plant inventory establishes a vegetational baseline for the site, providing the primary food base for the fauna identified in later analyses. The extraordinary richness of tree and shrub species in these sediments confirms that Kap Kobenhavn once supported a mixed ecosystem with no modern analog at this latitude. These findings are archived in the European Nucleotide Archive under accession PRJEB 55522, with all sequence data and taxonomic assignments available for independent review. DNA sequencing from the Kap Kobenhavn sediments produced a detailed inventory of small and medium-sized land animals inhabiting late Pliocene Greenland. Ancestral reindeer identified through mitochondrial DNA fragments indicate the presence of large herbivores adapted to forested environments at 78 degrees north. Reeds assigned to lemmings and hares confirm a diverse rodent and lagomorph community, both key prey species in modern Arctic food webs. The environmental DNA also revealed resident waterfowl, including ancient relatives of geese, suggesting the existence of open water bodies and seasonal wetlands within the boreal forest mosaic. Vertebrate DNA reads were authenticated by cytosine deamination patterns and verified against negative controls, with sequences deposited under ENA accession PRJEP 55522. This detection of terrestrial birds and mammals, alongside aquatic and plant taxa, demonstrates a complete trophic structure, from primary producers to herbivores and omnivores. The faunal assemblage, absent from present-day Greenland, points to a stable, productive ecosystem supporting multiple vertebrate niches. The diversity of vertebrate reeds, spanning mammals and birds, establishes that the Kap Kobenhavn Formation preserved a functioning land ecosystem with multiple trophic layers, preceding the discovery of larger megafauna in the same sediments. In July 2012, the laboratory team at the University of Copenhagen screened bulk sediment samples from the Kap Kobenhavn Formation for vertebrate DNA. The first mastodon signal appeared as ultra-short mitochondrial fragments, each less than 50 base pairs, aligned to the mammoth genus. These reeds were absent from all negative controls and displayed characteristic cytosine deamination patterns, confirming an ancient origin. By November 2012, shotgun metagenomic sequencing produced enough coverage to place the Greenland mastodon on a deep basal branch of the Proboscidean family tree, distinct from younger North American Mammoth americanum. Independent resequencing and authentication in March 2013 at a separate Copenhagen laboratory replicated the result. The phylogenetic analysis revealed that mastodons once ranged at least as far north as 78 degrees latitude, extending their known habitat far beyond previous fossil records. No physical mastodon remains were found at Kap Kobenhavn. The evidence derives entirely from sedimentary environmental DNA. This molecular trace, recovered from permafrost-bound mineral grains, documents the presence of large browsers in a boreal forest ecosystem during the late Paleocene. The discovery, catalogued under European Nucleotide Archive accession PRJEB 55522, challenged assumptions about megafaunal distributions and forced a revision of Arctic biogeography. In an internal interview, a senior molecular biologist described the moment as astonishing. Mastodon DNA in Greenland was not on anyone's list of possibilities. The confirmation of mastodon presence set the stage for further investigation into the region's extreme Pliocene warmth and its impact on both terrestrial and marine life. Environmental DNA from the Kap Kobenhavn sediments includes clear marine signals. Sequencing identified horseshoe crab DNA, a coastal invertebrate now absent from Greenland. Horseshoe crabs today require warm, shallow waters, and their presence in late Pliocene sediments at 78 degrees north latitude indicates a coastal embayment with sustained high temperatures. Alongside this, DNA from warm water green algae, including genera typical of temperate estuaries, was recovered from multiple sediment horizons. These algae require summer surface temperatures significantly above freezing, supporting reconstructions of mean annual air temperatures 11 to 19 degrees Celsius higher than present. The combination of marine invertebrate and algal DNA, authenticated by cytosine deamination and matched to modern reference genomes, confirms that the Kap Kobenhavn site was not only a boreal forest, but also bordered a productive temperate shoreline. The marine DNA signatures appear in the same sediment layers as terrestrial plant and animal DNA, demonstrating a mixed terrestrial and marine ecosystem unique for this latitude. These findings provide independent evidence for a shallow bay environment and serve as biological proxies for extreme Arctic warmth during the late Pliocene, reinforcing proxy-based temperature estimates and setting up later analysis of model data mismatches. Proxy temperature reconstructions from Kap Kobenhavn present a stark anomaly. Clumped isotope thermometry of carbonate nodules indicates mean annual air temperatures between 11 and 19 degrees Celsius warmer than present, despite atmospheric carbon dioxide levels near 280 parts per million, comparable to pre-industrial values. Leaf wax hydrogen isotope ratios, delta D, from plant lipids independently confirm this warming, with reconstructed summer temperatures exceeding modern Arctic means by more than 10 degrees Celsius. Pollen-based transfer functions, calibrated against modern boreal forests, yield consistent results, placing the late Pliocene Kap Kobenhavn ecosystem in a thermal regime with no modern analog at 78 degrees north. These converging proxies are anchored to sediment layers dated by magnetostratigraphy and uranium-thorium methods, minimizing chronological uncertainty. When the Community Earth System model, CESM, is run with Pliocene boundary conditions and pre-industrial carbon dioxide, it fails to reproduce the full magnitude of the observed warming, underestimating Arctic temperatures by up to 15 degrees Celsius. This mismatch highlights a gap between model sensitivity and empirical proxy evidence, suggesting that key feedbacks such as vegetation, cloud cover, or ocean heat transport remain underrepresented in current climate simulations. The Kap Kobenhavn dataset, archived under ENA accession PRJEB 55522, now serves as a benchmark for evaluating high-latitude climate response to modest carbon dioxide forcing. The climate anomaly recorded at Kap Kobenhavn stands as a direct warning for the present. Proxy evidence shows Arctic temperatures rose by up to 19 degrees Celsius, while atmospheric carbon dioxide, remained near 280 parts per million, levels similar to those before industrialization. Current models from the Intergovernmental Panel on Climate Change project strong polar amplification, but Pliocene data reveal that feedbacks, such as shifts in vegetation, cloud cover, or ocean heat transport, can drive even greater warming than models predict. This mismatch highlights the risk that modern climate projections may underestimate future Arctic change. The ancient DNA from these sediments also preserves genetic variants, or alleles, that enabled plants and animals to survive extreme warmth at high latitudes. Researchers now study these ancient adaptive alleles to understand resilience mechanisms that could help modern crops and wild species withstand rapid warming. The Kap Kobenhavn record demonstrates that Arctic ecosystems can reorganize under rapid climate shifts, but also that many lineages disappear. The findings urge a focus on genetic diversity and resilience breeding as climate policy priorities, using lessons from deep time to inform adaptation strategies today. The Kap Kobenhavn project ran for 15 years, adapting to new technologies and expanding its ambitions as results accumulated. The Lundbeck Foundation provided the initial grant in 2006, followed by 2 further rounds of funding in 2011 and 2015, totaling over 12 million Danish kroner. These grants enabled the purchase of a dedicated cleanroom facility in Copenhagen and the recruitment of specialist staff for bioinformatics and contamination control. Securing permits for fieldwork in Greenland required coordination with Danish and Greenlandic authorities, and future drilling in Canada and Russia now faces even stricter environmental and cultural heritage reviews. The team implemented rigorous chain of custody protocols and invested in negative pressure cleanrooms to meet evolving standards for ultra-low DNA contamination. The project's workflow and data management practices have since become a template for similar studies in permafrost regions. International coverage, including a PBS Nova feature in 2024, brought the findings to a wide audience and increased scrutiny of ancient DNA claims. With the age record set at Kap Kobenhavn, researchers are now targeting even older permafrost sites in the Yukon and Siberia, supported by new exploratory grants. The scientific community continues to refine extraction protocols and authentication methods, aiming to push the temporal boundary for ancient DNA beyond 2 million years. Ancient DNA from Greenland's permafrost demonstrates that polar ecosystems can reorganize rapidly under extreme warming, even with pre-industrial carbon dioxide. Current Arctic temperatures now rise 4 times faster than the global average. Every new sediment core expands the genomic archive, and the margin for error in climate forecasting shrinks. The molecular record warns that adaptation has limits, and the pace of change today demands urgent, evidence-based response. Your insights matter. Share them in the comments below.